Hello, I am Bryan Mounce. I am an assistant professor in the Department of Microbiology and Immunology at Loyola University Chicago. I'm very thankful to be here. I'm really excited to tell you about polyamines and some of the work we do in my lab. And I hope to teach you a few things about cellular metabolism and how viruses use cellular metabolism to replicate. And they do this by behaving badly. When you think about a virus, it's got this beautiful structure. They have a lot of really interesting, yeah, gonna have to redo that slide already. Sorry about that. Okay. When you think about a virus, you might think about a lot of things like its structure, its genome, the different proteins that make up a structure. But one thing that I like to think about is how a virus really acts like a bandit. They take over a cell, they use all the resources within the cell to replicate, and they're very selfish in that way, just like a bandit robbing a bank would be. Now, there are a lot of bandits out there. Many viruses exist, and my laboratory studies a number of them, including Coxsackie virus, which is an enterovirus. It does not have an envelope, so it doesn't have that lipid membrane around it. Chikungunya virus, which is an alpha virus, as well as an arbovirus, meaning it's transmitted by mosquitoes. It has an envelope as well as a capsid. Zika virus, which is a flavivirus, also an arbovirus. Its envelope and encapsulated. And then another virus called Rift Valley fever virus, which is a Bunya virus, and it's got a unique genome that's negative sense, and it's enveloped as well. Now, within my lab, we use these different viruses to kind of compare and contrast how these viruses use cellular resources, as well as how they interact with the cell. So when you think about how these viruses are bandits, these viruses aren't after money. They're after cellular resources like amino acids, like nucleotides, like lipids. And one of the resources I'm really interested in and that my lab really focuses on is polyamines. Where did the viruses get all of these molecules? Well, that comes from cellular metabolism, shown here in this beautiful network. Now, you might get some flashbacks to your biochemistry class back when you were in undergrad or in grad school, when you had to memorize all the pathways like the TCA cycle, or what I'm going to focus on, the urea cycle. We're not going to go over cellular metabolism in great detail, and of course I don't expect you to memorize anything, but what I'm going to tell you about are a few of the dots that come directly off the urea cycle. And as I mentioned, one of the cellular resources that these viruses really like is polyamines. So what are polyamines? Well, polyamines are small aliphatic molecules. They're comprised of putrescine, spermidine, and spermine. They are simple carbon chains, and they have amino groups on the ends. And you can imagine that these amino groups are really important for these polyamines. At physiological pH, they're positively charged, and this really engenders them with their function. So you can imagine that these polyamines combine to negatively charge things and affect their function. So what are those functions that polyamines affect? Well, polyamines are involved in nucleotide metabolism, as well as nucleic acid function. They're also involved in lipid metabolism and membrane rigidity. Again, you can imagine the polyamines binding to negatively charged head groups of these lipids. And they're also really important for protein translation through this really unique pathway that I'll talk about later in the presentation. Now, polyamines are really abundant resources within the cell. They're found at concentrations up to 1 millimolar. So you can imagine that their synthesis is really highly regulated within the cell. And so I'll take you through this synthetic pathway. The polyamines putrescine, spermidine, and spermine are synthesized from an ornithine precursor. And ornithine is derived from the urea cycle. The gatekeeping enzyme for this process is ornithine decarboxylase, or ODC1. So now ODC1 is the gatekeeping enzyme. And you can imagine that if we want to manipulate polyamines within a cell, this would be the enzyme to target. And in fact, there's a wonderful inhibitor of this enzyme called DFMO, or difluoromethylornithine. DFMO is a really great drug that we use frequently within the laboratory. It's a suicide inhibitor of ODC1. It transfers a fluorine group to ODC1 and it inactivates it. Now, ODC1, when it's blocked, then can't work, and polyamines are depleted within the cell. Another great thing about DFMO is it's really not toxic to cells at all. And in fact, people can take DFMO. It's an FDA-approved drug. It's used for the treatment of trypanosomiasis. And it's also in a lot of clinical trials as an anti-cancer molecule. So, in fact, we can use it in the laboratory as a tool to study polyamines, but it's also used clinically to treat various diseases. So now what we want to do when we treat cells with DFMO is verify that our cells are depleted of polyamines. And we can do this by thin layer chromatography. Now, shown here is a thin layer chromatogram showing the different polyamines, putrescine, spermidine, and spermine. And you can see that these splotches that are the polyamines are separated by molecular weight. Now, when we treat with increasing doses of DFMO listed along the top, you can see that the polyamines are gradually depleted. Now, there is a bit of spermine that's remaining in the cells, but for the most part, putrescine and spermidine are absent when you treat with DFMO. So a few years ago, when I got really interested in polyamines and how they function within the cell, I was also really interested in terms of how polyamines might function in a virus infection. So I did a very simple experiment. Here, we're going to take one of the model viruses that we work with, chikungunya virus, and we're going to do an infection with and without polyamines. So first, I'll show you this data. This is a time course of infection of chikungunya virus, where you can see the time on the x-axis and the titer on the y-axis. And chikungunya virus replicates in a typical sigmoidal curve. Now, if we do that same infection, but with polyamine depleted cells that we've treated with DFMO, we see that the virus essentially fails to replicate. It seems that these polyamines are really important for the overall replication cycle of chikungunya virus. And what's really satisfying about these experiments is that we can also deplete polyamines, but then we can add them back exogenously. So if you add polyamines to cell culture, what happens is those polyamines are taken up by dedicated transporters, and those transporters allow polyamines to enter the cell and function like they normally would. And when you do this, the virus replication is completely rescued. So this really suggests that the functional part or the antiviral part of DFMO is polyamine depletion. So now when you see this data, you might think, okay, this is great in tissue culture. But one thing that we also did was look at this in several animal models. And I'm going to show you my favorite one now. This is a zebrafish that's been infected with Symbis virus on the left. In green is Symbis virus. Now, this is a virus that's really closely related to chikungunya virus. It's an alpha virus as well. And you can see that this fish is very bright green. Symbis virus is neurotropic, and essentially the entire nervous system has been taken over by this virus. Now, on the other hand, on the right of your screen, you'll see a zebrafish that has been swimming in DFMO for several days. And you can probably appreciate that Symbis virus infection is significantly restricted in this fish compared to its brother that was not swimming in DFMO. So this really suggests that polyamines are important for viruses, both in the context of tissue culture as well as in the context of an animal. Now, when you see this data, you might be thinking, that's really interesting, but really, how are polyamines functioning during virus infection? And so you might think back to some of the functions that I mentioned earlier in terms of how polyamines function within the cell. And really, these processes are very important for viruses. But what we have found over the last several years of investigating how viruses use polyamines is that they tend to use polyamines in different ways. And this includes things like affecting particle infectivity, connecting with other metabolic pathways, attaching to a cell, and even protease activity in the case of enteroviruses. And polyamines are also really important for polymerase activity as well as viral translation. So I'm not going to tell you about all of these different functions of polyamines today, but one I do want to focus on is how polyamines connect to other metabolic pathways and how this affects virus replication. And through that, I'll teach you a bit more about how polyamines function within the cell. So to do that, I'm going to go back to our general workflow where we've got the polyamine synthetic pathway. We block that with DFMO. We see a reduction in cellular polyamines. And here we're going to infect the cells with Coxsackie virus B3. So Coxsackie virus is an enterovirus, different from chikungunya virus, which is an alpha virus. And what we have found is that Coxsackie virus actually uses polyamines a little bit differently. So shown here is a growth curve of Coxsackie virus with the time on the x-axis, the viral titer on the y-axis, and again, we see that typical sigmoidal curve for Coxsackie virus infection. If we do the same experiment, but with polyamine depletion, we see that viral titers are significantly reduced. So in the case of Coxsackie virus, we wanted to know what's going on that could be affecting virus replication. And we took a very broad approach to this. We did transcriptomics, where we left cells untreated or we treated with DFMO and did RNA-seq. And shown here is a volcano plot with a fold change in gene expression as well as the p-value on the y-axis. And there are a few things that I want to point out to you. First of all, there's not a lot of change in gene expression. What you'll notice is the x-axis, the fold change, goes from 0 to 2. So we see changes in terms of fourfold induction or reduction in expression. So the changes in transcription are not big. There are a number of genes that do certainly change their expression, but there's not anything that's changing its expression drastically. The one counterexample to that could be the dot on the top right of the graph. And this is in fact one of the polyamine synthetic genes. So that was a nice control in our experiment. But nonetheless, we wanted to investigate what these different genes were. And one thing that we kept noticing within our data was a lot of these genes, some of which are shown on the right, are actually cholesterol synthetic genes. So I'll take you through the cholesterol pathway in just a second, but we're seeing changes in all the different enzymes that are involved in cholesterol synthesis. If we show this via a heat map, comparing Coxsackie virus infected cells to DFMO treated cells, all of this compared to untreated cells, of course, these are the different cholesterol synthetic genes. And you can see during Coxsackie infection, there's actually an upregulation in a number of these different genes. And in fact, with DFMO treatment, there's a downregulation in most of these genes. So this is very curious. It kind of sets up this battle between Coxsackie virus and polyamines for these different genes to be expressed during infection. So now, thinking about the cholesterol synthetic pathway, I mentioned at the beginning that some of these pathways might give you flashbacks to your biochemistry class, and I'm going to do that, give you some of those flashbacks again. Cholesterol is synthesized from hemimethylglutarate-CoA, which is turned into mevalonate by really the rate-limiting step HMG-CR, and then through several other pathways that we're not really going to talk about in detail, cholesterol is synthesized within the cell. And so when you think about how polyamine depletion is decreasing the expression of these genes, as we see from our RNA-seq, we expected that perhaps cholesterol within the cell might be depleted with polyamine depletion. And in fact, that's exactly what we saw. We measured cholesterol with a colorimetric kit. Shown here is the data there with DFMO increasing in concentration on the x-axis and the cholesterol abundance on the y-axis. You can see that cholesterol is significantly depleted as we treat with DFMO. So this was really interesting. It's nice to see that changes in gene expression for cholesterol seem to really affect cholesterol within those cells. And so we wanted to verify that these different genes are affected by polyamine depletion. So we measured via qRT-PCR, HMG-CR expression, which is significantly reduced, HMG-CS as well, and MVD. So we're really seeing this response in these cells to polyamine depletion at the transcriptional level. We also verified this by Western blot, and we can see that HMG-CR and MVD levels are significantly reduced with polyamine depletion. So to summarize at this point, polyamines are important for cholesterol synthesis as a whole, and at least gene expression and translation of a number of factors in cholesterol synthesis is reduced. And this is important for cholesterol synthesis, of course, and we believe that this is very important for Coxsackie virus infection. So now going back to our original volcano plot where we saw all these different genes, we were looking at what these genes might have in common. What could tie all of these different genes together that might really explain our phenotype? And so one thing we came across was that a lot of these genes are regulated by this protein called SREBP2. Now, SREBP2 binds to the sterol response element. So SREBP2 stands for sterol response element binding protein. So when SREBP2 binds to the SRE, this results in the transcription and eventual translation of many genes involved in cholesterol synthesis. So this is the way that the cell can regulate the amount of cholesterol that is synthesized within the cell. And it's responsive to sterols within the media or within our bodies. So this made us think, perhaps what we're seeing is an effect on all of these cholesterol synthesis genes because, in fact, SREBP2 is sensitive to polyamines. And so we did a simple experiment where we did increasing doses of DFMO, and we looked at SREBP2 expression. Shown here is at the transcriptional level. And we were kind of puzzled. There was no change in SREBP2 expression at the transcriptional level. However, when we did the Western blot for SREBP2, we in fact did see a significant reduction in the amount of protein compared to untreated cells. So at this point, I'll summarize again, saying that polyamines are important for SREBP2 translation, but not transcription. And this facilitates cholesterol synthesis and gene expression, resulting in cholesterol that facilitates virus infection. Now I'm going to take you back to the original polyamine pathway. Shown here again is ornithine being converted into putrescine, into spermidine, and spermine. Now there's a really unique offshoot of this pathway, and that is this modification of the cellular factor EIF5A. So EIF5A is conjugated to spermidine. This is the only protein within our cells that is conjugated to spermidine. And after one additional hydroxylation of that spermidine moiety, this is called hypusination. So hypusination is a very unique modification. It's found in a number of organisms, including in bacteria, in a slightly different context. But within our cells, this is the only protein that's hypusinated. So what does EIF5A do? Well, you can kind of imagine that it's an important factor for translation, as I mentioned at the very beginning. EIF5A is an elongation factor, despite being named an initiation factor, that we think is important for the translation of possibly hard to translate regions of proteins. So now what is that? EIF5A is really important for translation of certain hard to translate proteins. And what those hard to translate regions consist of are polyproline tracks. So shown here is an analysis of the different motifs within a protein that require EIF5A for translation. And you can see that many of these are diprolines. You can imagine when a protein is being translated off of the ribosome, 2 prolines in a row incorporates a kink into that nascent peptide that can be hard to exit from the ribosomal exit tunnel. And so what you can then imagine is perhaps one of our cholesterol synthetic genes or SREBP2 probably has one of these diproline motifs that's very important for its translation. And so when we take away polyamines, we take away this hypusination event, and that makes it harder to translate our factor. And in fact, if you look at the sequence of SREBP2 shown on the right of your screen, there are several diproline motifs within that protein. And so what we're really interested in is whether this hypusination event in particular is responsible for our phenotypes that we're observing. So again, spermidine is conjugated to EIF5A. This is then hydroxylated to make that hypusine modification. And we can even inhibit this process with 2 different inhibitors. GC7, which is a synthetic inhibitor that's very specific for one of the enzymes that's involved in the conjugation of spermidine to the EIF5A. And then there's also deferoprone, which is actually used clinically in the treatment of various diseases. This inhibits the second step, which is catalyzed by deoxyhypusine hydroxylase, which is important for that hydroxylation of spermidine. Now, when you inhibit the hypusination event through either GC7 or deferoprone, we see that there is a decrease in the amount of hypusine within the cell, shown on this Western blot. But you might notice right above that hypusine Western blot, we also see a decrease in SREBP2 protein translation. So this is really suggesting to us that this hypusination of EIF5A is what's really driving our phenotypes. Now, of course, we wanted to look back on some of our data to determine is this affecting the cholesterol within the cell. And so again, we measured the cholesterol within the cell with an increasing dose of GC7, as well as an increasing dose of deferoprone. And again, we saw this decrease in cholesterol abundance, suggesting that this hypusination event is really important for cholesterol synthesis, probably through SREBP2. And so with that, I would like to summarize all that we've gone over today. I hope I've been able to educate you a little bit about how polyamines work within the cell. Polyamines, specifically spermidine, are really important for this modification of EIF5A. So EIF5A works as a translational elongation factor, and it functions in the translation of many cellular proteins, but one of those that we've discovered is SREBP2. So SRABP2, the sterol response element binding protein, binds to the sterol response element, and this drives the transcription of several cholesterol synthetic genes. And these are the genes that we found in our volcano plot when we did RNA sequencing on our cells. And this is important for the translation of those factors and then the synthesis of cholesterol within those cells. So cholesterol synthesis is really important for virus infection, and this results in the propagation of viruses, including Coxsackie virus, which is the virus we use as a model system in this case. And so if I can leave you with anything that is important in this presentation, it's that viruses really love polyamines, but what's important is that our cells also really love polyamines. And what's great about polyamines is that they are really important for a number of different events within the cell and during virus infection. And we've been able to really understand how polyamines connect to different events within virus infection, but they also connect to different metabolic pathways in the cell. And so you can kind of think of them as having connections to many different pathways within the cell that we're just beginning to understand. And through this, we hope to be able to break up that relationship between viruses and polyamines. So it's just our cells living happily ever after with their polyamines. And so with that, I'd really like to thank the people that have been instrumental in this work, currently within my laboratory, as well as past students. So this has been primarily led by Mason Firpo, who is on the bottom left of your screen, who did all of the RNA sequencing and who has been instrumental in pushing this project forward. And also I have to thank Priya Shah and Maureen Petit at UC Davis, who have been really helpful with analyzing our data and helping us to understand RNA sequencing. And I'm always happy to interact with anyone on Twitter or through our website. And I look forward to seeing you all in person in the future.